Okay, chapter 14 on eukaryotic gene structure and RNA processing. Here's the central dogma of molecular biology, and we've worked through transcription. Now we have RNA molecules, but they're not ready to take part, in translation yet. They need to be processed before being shipped out of the nucleus into the cytoplasm. And most of what we'll be discussing in this chapter focuses on eukaryotes, not prokaryotes. First, we'll talk about gene structure in eukaryotes and intervening sequences or introns and post-transcriptional modifications particularly to the messenger RNA, and then transfer RNA structure modifications and ribosomal RNA structure and processing. So genes can be defined in different ways. An old definition was a sequence of DNA that codes for a polypeptide. Functional definition can be an inherited factor that determines a characteristic, but a more modern definition is an entire DNA sequence that's required to transcribe and code for an RNA molecule. This includes things like introns, promoter regions, and terminators as well. So prokaryotic genes exhibit collinearity. That means that the messenger RNA is the same length as the gene that it came from. So here we see the gene on top, double-stranded DNA there. Here we see the messenger RNA in the middle. It's divided into codons. Remember that codons are triplets of messenger RNA that code for specific amino acids. And then we see the polypeptide chain at the bottom. Now, we don't see this in eukaryotes. In eukaryotes, the functional messenger RNA is way smaller than the gene that it came from. We'll talk about why that is shortly. So, eukaryotic genes can be divided into exons, which are express sequences. These are spliced together in the mature RNA, and they contain the codons. And then introns are intervening sequences. And these are initially transcribed into RNA, but then cut out of the RNA transcript, so they do not appear in translation at all. So, introns, we don't really know what their function is, but they can be 60 or more per gene. They can range in size from 200 base pairs to over 50,000 base pairs. Very common in eukaryotes, very rare in bacterial genes, and they're present in messenger RNA genes, transfer RNA genes, and ribosomal RNA genes as well. So this shows the human interleukin gene. The exons in this gene code for only about 165 amino acids. The exons are in the darker green, and the introns are in the lighter green. As you can see, intron 1 there. Intron 2 is very large. There's 2,400 bases of it missing in the middle, just so it'll fit in the figure better. Same thing, intron 3 has 1,900 bases missing in the middle, so it'll fit in there. So anyway, in this gene, there's more intron DNA than there is exon DNA. And remember, it's the exons that will be spliced together. And here there's only 4 exons. Here's the mouse beta globin gene showing you 2 introns and 3 exons and the length of each in base pairs. So the intron DNA is probably outnumbers the exon DNA. Here's a couple of other genes, ovalbumin on the left, cytochrome B on the right, and you can see the final messenger RNAs down at the bottom there in light green are much, much smaller than the genes that they came from above them. Notice how tiny the exons are relative to the introns. Okay? So, in eukaryotes, the size and number of introns is directly related to the increasing organismal complexity. Yeast, which are single-cell fungi, have only a few short introns. Drosophila, fruit flies, have longer and more numerous introns. And in us vertebrates, our genes are interrupted by very long introns. And we can group introns in different ways. Group 1 and 2 introns are found in ribosomal RNAs and some other things. And then there are nuclear pre-messenger RNA introns and introns in transfer RNA as well. And over on the right shows you the mechanism used for getting rid of them or cutting them out, and we'll talk about that later. So messenger RNA, let's discuss its general structure first. Reading from left to right, there's a 5' UTR untranslated region, sometimes called the leader, and it contains a specific sequence in prokaryotes for ribosomal recognition. Then there's the protein coding region with the codons, and then a 3' UTR after the stop codon. And it affects the stability of the messenger RNA molecule, the 3' UTR trailer does. So here we see a picture of a messenger RNA. There's the 5' UTR on the left, 3' on the right, starts on stop codons, and then the protein coding region in the middle that contains the actual codons, excuse me. So what we'll be talking about in this chapter are post-transcriptional modifications to RNA. That is, what do we do to the RNA after it's been made, but before it can be used in translation. Pre-messenger RNAs get a 5' cap and a poly A tail, a 3' poly A tail on the other end of the molecule. And then, of course, intron removal needs to occur in all pre-messenger RNAs, pre-ribosomal RNAs, and pre-transfer RNAs. And this lists the post-transcriptional modifications. We're not going to talk about RNA editing, however, but we will talk about the first three. And this kind of shows all three of them occurring, the 5' cap, the poly A tail at the 3' end, and then intron removal. I guess here the introns are in red. That red and green I have really trouble distinguishing, being red-green colorblind and all. Okay, the 5' cap. It's a backwards methylated G nucleotide. It's added to pre-messenger RNAs of eukaryotes only. It occurs rapidly after transcription. It helps protect the molecule from degradation by nucleases in the cell. And it aids it in transport out of the nucleus into the cytoplasm where translation occurs. It's also important in ribosomal recognition. So these different molecules have to be able to recognize each other and come together for these processes to occur. And as we'll see, translation is a pretty complicated process. So most eukaryotic mRNAs have a 5' cap. This shows the steps involved in adding the 5' cap. I don't care that you learn all these steps. I just want you to realize that it's a multi-step process and know what the 5' cap is and what its function is. Okay, so here's a close-up of it again, a backwards methylated G nucleotide stuck on the 5' end of the untranslated region of the pre-messenger RNA. Now the other end of the molecule gets a 3' poly A tail. And this means anywhere from 50 to 250 A nucleotides are attached to this after transcription. So it's not encoded in the DNA. This process is called polyadenylation. Remember that many genes that are transcribed by RNA polymerase II, which transcribes messenger RNA genes, will continue transcribing well after the stop codon. And there may be up to 1,000 base pairs that are cut off that poly A tail. So that the poly A tail confers stability to the messenger RNA molecule. And also it helps aid in transport out of the cell nucleus into the cytoplasm. So here we see this consensus sequence, and downstream of that, the 3' UTR is cleaved, and then the poly A tail is added in a process called polyadenylation. So remember, consensus sequence is the most common DNA or RNA nucleotide sequence, and it's AAU AAA. Downstream is cut, and in us mammals, at least 6 different proteins are involved in adding the poly A tail. And so this shows you what's going on here, the consensus sequence, and then the cleavage site, and then polyadenylation, the adding of the poly A tail. Now we're starting to discuss intron removal, and we'll do messenger RNAs first. So a spliceosome is a molecular structure made of small nuclear RNA and small nuclear ribonucleoproteins. That's what RNP stands for. Kind of looks like a ribosome, and it cuts RNA out of, introns out of the pre-messenger RNA. And again, we don't know what the introns are for, but they're such a big part of genes, they must have some important function. Remember that exons will be spliced together and they contain the codons. So this white triangular shaped blob is the spliceosome, and then that's RNA snaking out of it. And several molecules come together to form the spliceosome, and they cut the one end of the intron and kind of wrap it around to form a loop that's called a lariat. And then as we can see in 6 there, the exons are spliced together. So again, you don't need to know all these molecules, just know basically what makes up a spliceosome from the previous slide and what their function is. And again, this shows you how we have got to remove the introns, splice the exons together for our functional messenger RNA. And there are consensus sequences at the ends of the introns and exons, and they are important in intron removal. So here we can see what's going on here. The spliceosome cuts either end of the intron, bends one end around to attach it to the middle of the intron, forming this loop structure called a lariat that eventually gets degraded by the cell, and the 2 exons are spliced together in this instance. And that's all I've got on messenger RNA for now. Let's move on to discussing transfer RNA structure and processing. So, both bacterial and eukaryotic tRNAs undergo processing. Often several transfer RNAs are transcribed together as a large precursor tRNA, which is then cut up into the smaller functional tRNAs. And transfer RNAs, enzymes with endonuclease activity remove the introns. Remember that endonuclease activity is being able to cut the sugar phosphate backbone of the nucleic acid. So the job of transfer RNAs, remember, is to bring in amino acids to the ribosome during translation. These molecules are 74 to 95 base pairs long and have a cloverleaf shape, at least in 2-dimensional representations. And they can occur, tRNA genes, in clusters or scattered about the genome. There are many copies of them in eukaryotes in particular. They have an anticodon arm, which is complementary to the codon in the messenger RNA, a T arm for ribosomal recognition, another DHU arm, and then an acceptor arm, which is the 3' end of the molecule, and that's where the amino acid attaches. And to date, all sequenced have the sequence ACC at that 3' end or acceptor arm. So here's a model of a transfer RNA molecule. They also have some unusual bases that you don't find in other RNA or in DNA. Anyway, at the top, the 3', there's an AOH, that's where the amino acid attaches. The anticodon at the bottom there temporarily binds with the messenger RNA during translation, and that T arm is involved in ribosomal recognition. Another view of this, and the little red dots are the rare unusual bases. So you can see there's quite a few sprinkled around throughout the transfer RNA molecule. And so removal of introns then depends on enzymes in transfer RNA. So you can see there's the intron in the pink box, and it gets cut out with a splicing endonuclease, and then will be degraded, of course, and then a splicing ligase will come in and splice the rest of the molecule together, the exons. And that's how we process a transfer RNA molecule. Moving on to ribosomal RNA then. So we prokaryotes have a have a single ribosomal RNA gene that yields 3 different molecules, and there's only 1 copy of it per prokaryotic chromosome. The eukaryotes, though, have 2 different ribosomal RNA genes, a large gene that yields 3 ribosomal RNA molecules, and then a small one that yields a single small ribosomal RNA molecule. And they're often on different chromosomes. We can see that. And we have many copies of them. The large one we can see in fruit flies is 500 tandem copies. That means copies next to one another. They're in a line on their sex chromosomes. And then their small ribosomal RNA molecules on their chromosome 2. So Self-splicing then, this occurs in the removal of introns from ribosomal RNA. So here we say that the RNA is autocatalytic. When the RNA acts like an enzyme like this, it's referred to as a ribozyme. So this shows you the large ribosomal RNA genes. There's bacterial ones at the top there, just one copy. In the bean in the middle, Vicia faba is a bean plant. And you can see the 3 genes there. That thick black lines are called internal transcript spacers. So that's kind of like introns, if you will. And then ours down at the bottom. And of course, we eukaryotes have many, many hundreds of copies of these things because, of course, ribosomal RNA is used in making ribosomes. And ribosomes are used in protein synthesis, so they're very important. So yeast have between 100 and 200 copies. We've got about 280 copies of that thing I just showed you. And a species of frog is about 450 copies. So it just depends on what organism, how many copies they have of these ribosomal RNA genes. So this shows you a group 1 intron, which is very, very long and has complex secondary structure on the left there. And notice how the little exons 1 and 2 are just that darker green material from the red arrow to the end of the molecule. So they're tiny relative to the giant intron. But anyway, the introns, the RNA loops around on itself and splices the exons out and attaches them together. And that's self-splicing. And that's how ribosomal RNA molecules get rid of their introns. And so when these ribosomal RNA genes are transcribed, we get a long, big RNA molecule. The parts that are going to be kept are largely methylated, as shown by these little red lines sticking down, and the stuff in between is degraded. And so we can see the intermediates, and then at the bottom we get mature ribosomal RNA is a 16S RNA, 23S ribosomal RNA, and a 5S ribosomal RNA. And then in this case, a single transfer RNA molecule is in there as well. And in eukaryotes, it's similar. Our ribosomal RNAs are a bit larger, 18S, 28S, and 5.8S. S is Fedberg units, a way of measuring the size of molecules using a centrifuge. These are the karyotypes of 3 species of conifer trees, 2 spruce trees with 12 chromosomes, and a Douglas fir 13 chromosomes. But I want to draw attention to the little sort of bluish purple regions on those chromosomes, and those are the sites for the ribosomal RNA genes. So they're present on many chromosomes in certain species, and on others they're only packed together on 1 chromosome. The prokaryotic ribosome, of course, has a large and small subunit, and this shows you what ribosomal RNA molecules and proteins are involved in each subunit. Remember, these subunits stay apart unless translation is occurring. And these Fedberg units are not additive, so the whole ribosome is 70S. Our ribosomal subunits are assembled in our nuclei, and so the proteins are brought from the cytoplasm into the nuclei and combined with the ribosomal RNAs to make the ribosomes. This shows the components of bacterial and eukaryotic ribosomes. And so ours are 80S and bacteria are 70S, and we have more proteins and bigger RNAs. Although that 5S RNA is the same size in prokaryotes and eukaryotes. Interestingly, this shows a picture of a nucleus, a cell nucleus with the nucleolus. And the nucleolus is where the ribosomal RNA genes and molecules are, and that is where the ribosomal subunits are assembled in the nucleolus of the nucleus of the cell, and then those subunits are shipped back out into the cytoplasm where they can take place in translation. So, summarizing intron removal then, self-splicing or autocatalytic is how ribosomal RNA gets rid of their introns, enzyme-mediated for transfer RNA, and then the spliceosome for messenger RNA molecules. And that's that for chapter 14.